Hi, I'd like to thank the organizers for the opportunity to present my ACM-BCB 2020 proceedings talk, Predicting Protein Secondary Structure by an Ensemble Through Feature-Based Accuracy Estimation. This work was done by Spencer Krieger and John Kececioglu. I'm Spencer Krieger, and I'm excited to share this work with you today. The task we address in this talk is protein secondary structure prediction. Protein secondary structure is a local and discrete form of protein structure. Given an amino acid sequence, each amino acid is assigned one of 3 or 8 secondary structure classes, depending on the model. The task is, given amino acid sequence P, predict the corresponding secondary structure sequence S of the folded protein. For each amino acid position in P, S gives the corresponding secondary structure state, where in the 3-state model, E is beta strand, C is coil, and H is helix. There are many tools currently in use for protein secondary structure prediction, some of which do both 3 and 8-state prediction, while others only predict on the simpler 3-state model. All of these tools, except NESSY, perform a ssyBLAST search to find homologous protein sequences from which a position-specific scoring matrix for the input amino acid sequence is computed, that is then input to a machine learning model. JPRED, SciPRED, SSPro, and Porter use various forms of neural networks. DeepCNF uses conditional neural fields, and PSRSM uses a form of support vector machines. NESSY forgoes the ssyBLAST search and instead uses metric space nearest neighbor search over a smaller template database of fixed-length amino acid words, followed by a dynamic programming algorithm to compute a maximum likelihood physically valid structure prediction. Ensemble methods are an attractive approach for machine learning that can take advantage of such a collection of state-of-the-art tools. Ensemble methods in general take multiple independent predictions as input and produce one final prediction output by the ensemble. In particular, these predictions may be combined by voting, which can include weights on the votes, averaging, which is usually applied to real valued input, or by stacking, where a final predictor is trained over the input predictions to yield the prediction output by the ensemble. We can categorize ensembles into 2 types. Homogeneous ensembles consist of predictors from the same machine learning model class. Most secondary structure prediction tools internally use a homogeneous ensemble trained on different subsets of the data called bagging, which are then combined using stacking. Heterogeneous ensembles, on the other hand, combine predictions from different model classes. There's much less work in the literature on homogeneous ensembles for secondary structure prediction. Prior heterogeneous ensembles all contain models that they internally trained themselves. In contrast, our work presents the first ensemble method for secondary structure prediction that combines predictions from independent tools from other developers. We evaluate the accuracy of predictions using the standard measure of Q3 or Q8 accuracy, which is the fraction of residues in the protein that are correctly predicted under the 3 or 8-state model. The combining methods of prior heterogeneous ensembles only achieve an improvement with an order of magnitude less than 1% Q3 accuracy over straightforward averaging, and in the end only achieve an improvement of around 1% over the best single predictor in their ensemble. For combining predictions in our work, we use the technique of feature-based accuracy estimation, which was originally developed by de Blasio and Kececioglu for parameter advising for protein multiple sequence alignment. We recently applied this technique for the first time to protein secondary structure prediction at ISMB 2020. In general, accuracy estimators map real-valued features of a prediction into an estimate of the unknown true accuracy of the prediction. In particular, a prediction method outputs a prediction from which features are extracted that are input into an accuracy estimator that produces an estimated accuracy. To preview our contributions, our new ensemble approach exceeds the accuracy of the best state-of-the-art tool by nearly 4% under the conventional 3-state model, and by more than 8% under the challenging 8-state model. For our ensemble approach, we developed feature-based accuracy estimators for state-of-the-art tools where the average error of these estimators ranges from 5 to 7% for the 3-state model and from 6 to 11% for the 8-state model. Our ensemble outperforms the accuracy of simple voting by more than 2% for 3 states and nearly 5% for 8 states. A unique aspect of our ensemble approach is that as the number of predictors in the ensemble increases, the ensemble accuracy does not degrade, even as low accuracy predictors are added. We perform a thorough study of 7 ensemble variants, which differ in how their predictions are combined. These variants can be categorized into 2 groups: those that use per residue voting, which piece together different input predictions at each residue to form the final output prediction, and per protein selection variants, which select one input prediction over the entire protein as the final prediction output by the ensemble. In more detail, per residue voting variants vote on the secondary structure state output by the ensemble at each residue of the protein. In uniform voting, each prediction gives a vote of weight 1 for its predicted secondary structure state. The figure shows 3 input predictions that are combined into one output prediction by uniform voting. In the highlighted box, 2 out of the 3 predictors agree on their state yielding the output prediction CHE, which occurs in none of the input predictions. Further, we have confidence voting, which weights these votes by the confidence that each tool has in its predicted state, and estimator voting, which weights each vote by the estimated accuracy of each tool's prediction, which will require that we develop an accuracy estimator for each tool. On the other hand, per protein selection variants apply a scoring function to each input prediction and choose the one prediction over the entire protein that has the highest score, as seen below. In support selection, the score of the prediction is the total vote for its states from other input predictions using uniform voting. In confidence selection, the score is the confidence of the tool in its prediction, averaged across the protein. And in estimator selection, the score is the estimated accuracy of the tool's prediction under its estimator. The last per protein selection variant is the hybrid approach, which combines a core prediction method with an alternate prediction method. Given the core and alternate predictions, we estimate the accuracy of the core prediction. Using a threshold tau, learned when training the hybrid approach, if the estimated accuracy is above the threshold, the hybrid outputs the core prediction. Otherwise, it outputs the alternate prediction. This discerns when to switch between the core and alternate methods based on estimated accuracy. Note this also only requires an accuracy estimator for the core method. Many of these variants use accuracy estimation. Recall that an accuracy estimator takes in real-valued features. In our approach to accuracy estimation, the first step is to map each feature separately to an initial accuracy estimate. These 2 figures show 2 different features. In the scatter plots, the horizontal axis is the feature, and the vertical axis is true accuracy. Each point represents a protein or a residue of a protein from a benchmark dataset for which true accuracy is known. Notice the nonlinear trend of true accuracy versus feature value. To address this, we obtain the initial accuracy estimate by fitting a piecewise linear mapping function for each feature. For NESI, the 2 features we use are template similarity which reflects how closely a protein sequence matches the template database, and Nessie's confidence in its prediction. And these two plots are the two plots shown below. In the second step, we combine these initial estimates into a final estimated accuracy. These two figures plot true accuracy versus estimated accuracy for the two features of Nessie, where the dotted line is the identity function, and the closer a point is to the dotted line, the better the estimated accuracy. To combine these initial estimators, we use linear regression where we fit to true accuracy. When the features are independent, the final estimator has less noise than the initial estimators, as illustrated in the figure by the closer trend to the dotted line on the right. We now present our experimental results. We evaluated the state-of-the-art tools and ensemble variants on CASP and PDB benchmark datasets, which contain over 900 proteins in total. The PDB datasets take a random subset of proteins from PDB for each year between 2016 and 2019. For the template-based tool Nessie, we use a template database formed only from the proteins available prior to the date of the CASP competition or the year of the PDB dataset. We learn a separate accuracy estimator for each tool, where these accuracy estimators were fit on all of the benchmark proteins, which is essentially equivalent to leave one out testing, as all estimators have at most 4 parameters to fit. For each ensemble variant, we found the optimal subset of tools over all possible subsets for the ensemble that gave the highest average accuracy for the variant. This table lists the ensemble variants and their Q3 accuracy for the 3-state model, averaged over the CASP and PDB datasets. The accuracies are colored from red to green with increasing accuracy. Hybrid selection is the best ensemble variant and gives around a 2% accuracy boost over uniform voting. With respect to Q8 accuracy, which is shown in the right column of the table for the 8-state model, hybrid selection is again the best variant and gives nearly a 5% accuracy boost over uniform voting. Among other variants, estimator selection is the second best and is especially close in Q3 accuracy, where it gets within 1% of the hybrid selection. Here we compare the accuracy of state-of-the-art tools to the best ensemble, which is hybrid selection. Again, the accuracies are colored from red to green. And hybrid selection exceeds the accuracy of the best single tool by nearly 4% for Q3 accuracy. Once again, looking at Q8 accuracy, hybrid selection exceeds the accuracy of the best tool by more than 8%. Here we show the tools that comprise the optimal ensemble for each variant. For the 3-state model, the optimal estimator selection ensemble uses every state-of-the-art tool except JPRED, while for the optimal hybrid selection ensemble, the core method is Nessie and the alternate method is Porter. For the 8-state model, the optimal estimator selection ensemble uses every state-of-the-art tool except for DeepCNF. And once again, for the optimal hybrid selection ensemble, the core method is Nessie with an alternate method of Porter. We found the optimal subset of tools at each cardinality for each variant. Note that at higher cardinalities, this adds less accurate tools to the ensembles. The figure shows the Q3 and Q8 accuracy for increasing ensemble cardinality with Q3 above and Q8 below. We plot the accuracies for the two best ensemble variants with hybrid selection in red and estimator selection in green. Notice that these methods roughly improve or remain stable as cardinality increases. We now show the curves for the other ensemble variants, including per residue voting methods. While the hybrid selection and estimator selection ensembles remained stable in accuracy as ensemble cardinality increases, the other variants tend to degrade at higher cardinality. To study the limit on accuracy that can be achieved with current tools, we consider an oracle ensemble that optimally combines predictions using unknown true accuracy, which cannot actually be achieved in practice. Note, however, that with perfect accuracy estimators, which return the true accuracy of a prediction, estimator selection is equivalent to an oracle ensemble. With our current estimators and current state-of-the-art tools, estimator selection comes within around 2% of the oracle limit for the 3-state model and within 3% for the 8-state model. Here we study the error in accuracy estimators. We show histograms giving the empirical distribution of the error in accuracy estimators for each state-of-the-art tool, which is the difference between estimated and true accuracy. Note that the histograms are centered roughly around 0 and have a bell-shaped curve. To be able to investigate the effect of decreasing error, we fit a normal distribution to the error histogram for each tool as illustrated on the right. This enables us to simulate reduced estimator error by decreasing the standard deviation of the normal distribution. Using the normal distribution of error for each estimator, for each tool's prediction, we randomly sample an error value from the distribution and add it to the true accuracy, giving a simulated estimated accuracy for the structure prediction. These simulated estimated accuracies are then used by the ensemble variants. The figure shows the simulated Q3 accuracy of an ensemble as the standard deviation in estimator error varies across the horizontal axis. We vary the ratio of the standard deviation of the simulated error divided by the standard deviation of actual error, which is applied to the error distribution of all tools. The curve for the average simulated accuracy of the estimator selection ensemble is shown in black, while the curve for the hybrid selection ensemble is in blue. The dashed lines give the accuracies of the estimator selection oracle containing all tools in black, and the hybrid selection oracle containing only Nesi and Porter in blue. Note that as the standard deviation ratio goes to 0, the curves approach the oracle limits. The figure shows that decreasing the standard deviation of estimator error increases the simulated ensemble accuracy. We observe in practice that by adding a second feature to Nesi's accuracy estimator, we saw a 19% decrease in its standard deviation. Accordingly, by discovering more features that are relevant for accuracy estimation, estimator selection could exceed the accuracy of hybrid selection in practice, as suggested by the simulation, whereas currently estimator selection is slightly below hybrid selection. To conclude, our new approach to ensemble protein secondary structure prediction exceeds the state-of-the-art accuracy by nearly 4% and by more than 8% for the 3- and 8-state models. Our current ensemble approach leverages the new technique of feature-based accuracy estimation. Currently, hybrid selection is the most accurate ensemble method and is easy to implement, requiring only 2 prediction methods and an accuracy estimator for only one of those methods. Simulations suggest that estimator selection could surpass hybrid selection if estimator error is reduced, which may be achieved by discovering new features for accuracy estimators in the future. This work on ensemble prediction is implemented in a new tool called ssylla, short for Protein Secondary Structure Prediction, by an ensemble leveraging accuracy estimation. ssylla includes accuracy estimators for the state-of-the-art tools in the study and an implementation of the hybrid selection ensemble variant. Source code for ssylla is available free for non-commercial use at ssylla.cs.arizona.edu. We would like to thank the U.S. National Science Foundation for supporting this research. Thank you for listening to my presentation.